Welcome back to Biominds, your guide to mastering molecular biology and launching a high growth career in the life sciences. If you're new here, make sure to hit that subscribe button so you never miss a lecture. Let's get started. So today we will learn about compound microscope. So microscope is the one of the great invention of the science, as microscope can help in the finding or observing the little things which are surrounding us, and it is very important in the microbiology or in the biotechnology, in the molecular biology, etc. So we will learn about the compound microscope, which is the very common microscope we use in the labs or in the college labs as well. Okay, so compound microscope and the light microscope, so basically these two are the same. So we need something, we need light to observe the specimen, right? So we use the light microscope and the compound microscope, so together we have the compound light microscope. So this light microscope refers to the use of any kind of microscope that uses visible light to observe the specimens. So we have the visible spectra. Okay, so we use in the college also we use the visible light as a light source for the compound microscope. So many labs we use the sunlight, okay, as a uh, light, the visible light to observe the specimen as a light source. So basically light microscope and the compound microscope will get the compound light microscope. So compound light microscope means it's the microscope where we use the visible light as a light source. So if you see here, sometimes there are certain microscope that uses the artificial light, that is also visible light, basically artificial light or a sunlight, that is the natural light. Many colleges we use uh, as natural sunlight as a light source. Okay, or there are certain microscope that uses the artificial light as a visible source. Okay, so this is the compound light microscope. This is their diagram. So we have certain uh, you can see there are certain parts of the microscope. So we have, from starting from here, from the light source we can see. So here we have the illuminator means that is the light source that illuminate. Next we have the condenser. The important parts will see the stage, objective lens, arm that we use to hold, body tube, and the ocular lens. So here the important parts are the illuminator. Okay, so illuminator is where we get the light source. So it can be your artificial light source, or it can be your natural sunlight. Next we have the condenser. So here we have the condenser that actually helps in the focusing the light, because light can be sprayed. Okay, so we don't want light to spread more, so the condenser condense the light at the base. So here you can see the stage. Okay, so stage where we keep our specimen, okay, the things which we wanted to observe. Okay, next we have the objective lens. Can you see this? These are the objective lens where the lens, when we use in the cameras, the lens which focuses, the lens the capture that captures the images. So here also this lens captures the image of the specimen. That means you can see it, observe it. Okay, so we have the objective lens. This is arm. So arm, it is used to hold the microscope. Okay, next we have, this is the next part which is important, the ocular lens, which is there here. From here we can observe, that's what's the ocular eye, also known as the eyepiece. Okay, from where we observe the specimen. Okay, so this is the basic parts of the compound light microscope. So here is the image. You can see how the light passes. So from the illuminator, the light ray passes to the condenser. So condenser, actually here you can see the condenser lenses which actually the direct the lights towards the one point at the specimen. Can you see? Yeah. So next is the objective lens, from where the uh, you can get the magnification. And next is you have the ocular lens. Okay, so this is the basic part. Next comes the magnification. Magnification means how much you can zoom, okay, how much you can able to observe. So it depends upon the what specimen you want to observe. If it is a microorganisms, if its size is very low, then you need a high magnification, means you need something that focuses more. Okay, if you wanted to observe certain things which is larger than the microorganisms, then you need a low magnification power. So total magnification is the something that of a specimen by multiplying the objective lens magnification by the ocular lens magnification. So we have the objective lens. So in the objective lens we have three different lenses: 10x, which is the low power; 40x, which is high power; and 100x, which is also high power, but it is an oil immersion. Okay, and ocular lens has only one power, that is 10x here. So multiplying by this, you will get the total magnification of the microscope. Okay, so it helps in the zooming of the of things. It depends upon the which things you are observing. Okay, next comes the term resolution. So resolution is also known as the resolving power. So it's the ability of lenses to distinguish fine details and structure. So see, you just don't need to just observe the things, you need to observe the specimen very clearly. If you wanted to observe certain microorganisms, so you need to observe its structure, right? How it is, the shape of the microorganism, and the detailing of the microorganisms to study it. Okay, so resolution is, or the resolving power helps the microscope to analyze the fine structure of the things or the microorganisms. Okay, so if the microorganism, it refers to ability of lenses distinguish two points a specific distance apart. See, if a microscope has a resolving power of zero point 0.4 nanometer, it can distinguish two points if they are at least zero point 0.4 nanometer apart. Means, suppose there is a microorganisms and the microorganisms are very small, and if it two microorganisms, the distance between the two microorganisms is zero point 0.4 nm. Okay, and you wanted to observe the single microorganisms, so it should have the resolving power of zero point 0.4 nm. So only you can get the distance, or you can see the uh, microorganisms very clearly. Okay, light white light used in the compound light microscope has a relatively long wavelength and cannot resolve structure smaller than about zero point 0.2 micrometer. So see, the shorter the wavelength of light used in the instrument, the greater the resolution. Okay, so here we use the visible light. Okay, so if you want a more resolution, means if you want to observe certain things which are very very small, the smaller than zero point 0.2 micrometer, then you need certain other microscope. We have different types of microscope. Okay, next is, so here you can see the image. So we have the unaided eye, that is naked eye, you can see the tick. Okay, next we have the light microscope. So here you can see this images. But if you want to observe the structure in detail, it's you need the more advanced type of microscopes. Okay, next important term comes in the light microscope is the refractive index. Okay, so refractive index is something, it measures the light bending ability of the medium. So see, there are certain microorganisms which are transparent. So if you put it into the microscope, you will not able to see it. See, if you made a certain slides as well, and if you use the white background, and your font is also very light color, yellow color, so you cannot able to see the font clearly. Okay, so here if you see this slide, its background is dark, and its font is light, so that's why you can observe, you can clearly read the statements or the sentences. So like that only, the refractive index is very important. It measures the light bending microscope bending ability of the medium. So how we can do that? So we actually stain them, we actually color them. Okay, so staining is also one of the important uh, part of the microbiology. Okay, we will have the detailed session on this staining. So basically after staining you can get the contrast images. Okay, and next is for 100x magnification, highest magnification, we use oil. So why we use oil? Basically we use cedar wood oil. So this oil actually helps uh, so this immersion oil has the same refractive index as a glass. Okay, so the oil becomes part of the optics of the glass of the microscope. Okay, so unless the immersion oil is used, light rays are refracted as they enter the air from the slide, and the objective lens would have to be increased in diameter to capture the most of them. But we can't do that. We cannot increase the diameter of the objective lens to capture the refracted rays. Okay, so that's why we use oil, so that refracted rays can be captured by the objective lens. Okay, so if oil is not used with an oil immersion objective lens, oil immersion objective lens is 100x. The image becomes fuzzy with poor resolution. So here you can see the detailed diagram. So here we have the light sources and the condenser. So here you can see we use the oil. Okay, the oil we use, it is on the specimen. Okay, between the objective lens and the stage. So oil, you can see this blue light is getting into the objective lens, but this red, if it is, if here we do not use oil, so it goes out of the objective lens, so you cannot see the image clearly. Okay, you will see fuzzy image. Okay, to observe it clearly we have to use oil. Okay. So this is the basic diagram of the my compound light microscope. So these are the important parts of the microscope, important principle behind the compound microscope. Okay, so I hope, so this is the reference. So thank you. So we'll cover most of the staining and other topics in the next lecture. Thank you.